Okay. And our final speaker before lunch this morning is going to be, please excuse my pronunciation, Peter Theil from Aarhus University in Denmark.
Thank you very much. I hope you can hear me.
We can hear you and we can see your slides.
That's great. Thank you. Thank you very much. And thank you for your invitation and the opportunity to give a talk here. And I'm really sorry I couldn't make the trip to the US. Yeah, I hope it will be better in the near future. So I'll be talking about the colostrum production in sows and how it's produced, and yeah, a lot of things related with colostrum production in the sows. Just initially, I would like to consider cross species. Why is colostrum so important, and why have we focused on immunoglobulins? Mainly from the very beginning, I think transferring immunity is highly essential, and for pigs, it's really important because the pig is not able to transfer immunity across the placental membrane, and the outer circle here represents the amount, the size, or the area represents the amount of immunoglobulins in colostrum, whereas the inner circle represents the amount in the milk. And comparing just the pig with the human, you can see a huge difference. The pig has a lot of IgG because they need to transfer immunity through colostrum, whereas human can transfer that to the unborn babies during the fetal stage. So moving on to the sows, they give priority to different traits as gestation and lactation progresses. Initially, they prioritize fetal growth. Then more colostrum or colostrum becomes more important. Farrowing process, milk yield after farrowing, and at the end of lactation, future offspring starts to become important. So going just a few years back in time, we would clearly say, well, colostrum is being produced in late gestation before sows start, or it will stop when sow starts to build nest. And then when the first piglet is being born, the uterine contractions are expelling the piglets, then the colostrum has been produced. That was our way of thinking, and we have this stage 1 and stage 2 of lactogenesis. We believe it's quite different today. So we actually believe that most of the colostrum is being produced after the first piglet is being born, and I'll hope to convince you today how we came to that conclusion. So just initially, what is colostrum? Well, it's the first secretor from the mammary gland. It is being secreted from the first piglet is being born until at least 23 hours later. The latest we have seen, where if we consider stage 2 of lactogenesis, the copious milk production, and that is 39 hours after onset of farrowing. But to keep it simple, we consider mammary secretions from 0 to 24 hours after the first piglet is born as the mammary as the colostrum. And this is not completely correct, but it's just very easy if we want to quantify it, and if we want to take samples and so on, it's easy for us. You can see it's a gradual change. We see the piglets grow initially, then the piglet growth levels off. It seems like colostrum is being more or less emptied from the mammary gland, and then suddenly we have an increase in the rate of gain again. If we consider the concentrations of immunoglobulins from secreted or colostrum samples, we can see a very rapid decline, and it makes probably good sense to say, well, 24 hours is roughly the colostrum. Just for those that work with dairy cows, I want to say it's so much easier working with dairy cows than the sows, because everything is a big mess. It's really difficult to measure. We cannot measure it directly, the amount. It's hard to take a sample. It's working with colostrum studies. difficult to predict when is the farrowing starting and also when it ends, because we have so many piglets. Even when we see a placenta being born, we don't know that it's ended yet, because there are 2 uterine horns and so on. We have a lot of variation to cope with: litter size, birth weight, suckling stimulus of the piglets, and yeah, the farrowing length. We also see a decline in the colostrum intake per suckling bout, and it's tough to do the hand milking. And most of these farrowings we are studying, they are actually occurring in the nighttime. So it's really not easy to study these kind of things. I tend to consider my piglets as milking machines. They differ in number, they differ in size, they grow, they compete with each other, they have different stimulus, and they decide how often colostrum should be removed. So it's definitely easier to work with dairy cows. Just to show how colostrum intake change over time. The first meal is way bigger than the second meal, which is again bigger than the third meal, and so on, simply because in the very first meal, then you have an empty stomach, you have an empty intestine, so the piglet is able to ingest more at the first time and then continuously less amount. So if we really want to understand how much colostrum a piglet is ingesting and also how much a sow is producing, we would switch to the deuterium oxide dilution technique, which is also called heavy water. Just want briefly to mention how this is done, so you have an idea of that we can measure it pretty precisely. So we take a piglet, take a blood sample to measure the background of the deuterium. Then we take a syringe, fill it with a 10% solution of deuterium, and infuse that into the pig. And the initial dilution from the syringe into the concentration in the piglet after the isotope has equilibrated, that is used to calculate the body water. So now we know how many grams of water that piglet contained initially. And then we put the pig back to the sow and let it suckle colostrum until 24 hours after the first piglet was born. And the second dilution is due to the water intake from colostrum, and thereby we can measure actually how much colostrum has been ingested. We did that, and we made a model to make a prediction model, because this is really heavy work. It's also extremely costly, so this is not something we're doing over and over again. We wanted to have a model that we could use to measure it fairly easily, and we built that model, tried to understand if there was any impact of outdoor temperature or weather conditions on the ventilation system, whether the composition of colostrum had any impact, and whether sow characteristics had any impact. Our final model revealed that we should only consider the body weight at birth of the piglets, the weight gain in the colostrum period, and the duration in minutes for each piglet that could suckle colostrum. And then there was weight gain per birth weight and weight gain divided by duration. And all we wanted to measure was the colostrum intake of individual piglets, which we then can sum and quantify the colostrum yield of the sows. And you can see on the figure here, colostrum intake is highly linearly positively related with the weight gain. This is the most important predictor, actually. So what's the importance of the colostrum? Well, it's really highly important for the survival of these piglets. They are vulnerable. They don't have any fat actually at the body. They are losing temperature, and they need energy just to stay alive the first 1 or 2 days. So Hélène Canel and co-workers from INRA in France, they figured out that 250 grams seems to be a critical lower limit to ensure a good chance of surviving. This is really a challenge for us. We have been selecting the pig industry, not only in Denmark, but in various companies worldwide. They have been selecting for high prolific sows, which has really challenged the productivity. The farrowing length has increased, the colostrum yield needs to be improved. The colostrum intake is really compromised, and the piglet survival. And this is what we really have been looking into to improve by nutritional means, for instance. I'll not talk much about nutrition today, but this is clearly a hot topic for me as well in my research. So as litter size increases, what happens to the colostrum yield? Well, initially it increases until approximately 15 piglets in the litter. Initially, I would say the limiting factor of sow colostrum yield, that will be the removal of the piglets. The litter, the size of the stomachs in these piglets, that is actually the thing that is limiting the colostrum yield of the sows. But when we reach 15 piglets and above that, it seems like we reach a plateau, and now we have a situation where the colostrum production is limited by maybe the genetics, and definitely also by the nutrition of the sows. And this is why we really want to improve the nutrition. Could be nice also to improve the genetic trait or the selection, but that's really tough and not easy when we talk about colostrum. If we switch to colostrum intake of the piglets, we can see it steadily decreases, but from 15 and onwards, it goes pretty fast, and that is where we have more piglets than we have mammary glands. So it makes good sense that it's now going pretty fast down, and this is becoming really serious in the future, I believe, if we continue selecting for high prolificacy. Just to illustrate the distribution of sow colostrum yield, we have a mean of 6.1 kilogram, and nicely normally distributed, as you can see here. We have a problem. Approximately 7% of all our sows, they don't produce enough colostrum to actually supply sufficient colostrum for even the medium piglets. So these sows in the low range here, they are really challenged, or the litter is really challenged. We consider the colostrum intake of the piglets, it's also nicely normally distributed. We have a mean now of 328. 12 or 13 years ago, it was above 400 grams. So this selection for high prolificacy has really challenged the piglets. And you can see today we actually have a situation where approximately 30% of the piglets ingest insufficient amount of colostrum. So they are in a high risk of dying simply because of this low colostrum amount. So it makes really good sense that we are trying to do what we can to improve the colostrum yield and thereby the colostrum intake of the piglets. We did some studies some years ago trying to understand what is really affecting the mammary gland development, the mammary function of the tits. And we tried to tape some tits in the colostrum period to understand how fast will the milk production be initiated, how fast will it stop when they are not suckled. So we taped some glands. You can see over here what happens with a gland if it's not suckled. It completely regresses roughly within a week. So it goes pretty fast, this development. We had in one of our studies, we had 3 different suckling regimens. We had glands that were suckled normally by piglets, some that were transiently suckled during the colostrum period, and some that were not suckled at all. And we looked into the transcription of prolactin receptor and also of alpha-lactalbumin as an indicator of where they produced milk. And interestingly, those that were suckled 24 hours, now I'm talking only about the mammary biopsies collected at day 1. So these mammary gland has been suckled for up to 24 hours. When we took this biopsy, they had a high transcription of prolactin receptor, and they had a very high transcription rate of alpha-lactalbumin, indicating they are producing milk. Those that were only suckled 12-hour and then taped the next 12-hour before we took the biopsy, clearly had a lower transcription of prolactin receptor and consequently a lower transcription of lactalbumin. So very fast we see a shutting down of the prolactin receptor expression and thereby decoupling the prolactin signaling from the hormone. And consequently, we don't see much milk production simply because the pressure builds up, and yeah, that's the way it solves the problem if we have milk stasis. And even more pronounced if the gland has never been suckled, it's even lower expression of the prolactin receptor and also of the alpha-lactalbumin. So, and if we consider the next week, we don't see much difference. It actually happens within the very first day of lactation, this very fast change. So it's not many hours we have to do any changes if we want to, for instance, do some litter equalization. We want to understand when is colostogenesis taking place, because if we're going to do some nutritional manipulation, we have to understand when does it happen. The very first signs, if we consider the literature on when colostogenesis occurs, that is reported by Dodd and co-workers, and they reported that beta-lactoglobulin was the very first whey protein which can be found in sow plasma approximately 5 weeks before parturition. So approximately day 80 of gestation, we'll start to see the very first indication that the mammary gland are producing something which is coming from which is going into the colostrum. But it's definitely not much, and not until 3 to 4 days before parturition, lactose becomes detectable in the plasma because we have leaky tight junctions in the mammary gland, so it starts to be produced, but it leaks back to the plasma fraction of the blood, and then it can be detected. And we can see a very nice increase very late in gestation, indicating that the colostrum is being produced as the farrowing approaches. And this was actually also what we believed in for quite a long time. If we consider just visually when is it occurring the majority of the colostrum production, it could also support that, well, it happens in the late gestation. Day 105, we do not see much mammary development, and at day 114, really well developed mammary glands. So that indicates this is filled with colostrum. We then took some biopsies sent to Juan Lua's lab, and they did some really nice studies on gene expression or the whole transcriptome to understand what happens with the mammary glands. We took biopsies 14 days before parturition, and then every 4th day until after parturition. So we took also on the day after parturition at the end of the colostrum period. And what was evident was nothing really had happened compared to day -14. At -10, nothing had happened. At -6, nothing has happened. But then from -6 to -2, clearly a lot happened. And even more from -2 to +1. So it seems like all the genes are differentially, or those that change, they change from day -6 to day +1. So the last week, roughly, before parturition, this is where the mammary gland really change. And in that study, what were really upregulated, that was protein and carbohydrate metabolism, immune genes involved in the immune system, lipid metabolism, PPAR signaling pathway, prolactin signaling, and activating of some transcription regulators. That did not really tell, this does not really tell when the vast majority of the colostrum is being produced. It just tells something happens. But when is the real amount being produced? That was still what was puzzling me. We have done a lot of studies where we tried to feed differently in the last week before parturition, because we had to believe this is where it was being produced. So we should consider the last week of gestation and try to do the different feeding strategies or diet composition to improve the colostrum production. But we didn't really succeed very well on this. And then we started sorry, just a moment. Then we started to consider differently. We had to understand the nutrients. How are they flowing? So we did some studies and development, making some multi-catheterized sows. So we did some surgeries and put in catheters different places, so we could detect, for instance, how many nutrients was taken up across the GI tract, and the mammary gland, of course, is really important for us. So we were interested in understanding how many nutrients was taking up. And especially the glucose is important. And you know, glucose, when it circulates in the plasma, it's around 5 millimoles per liter. And the glucose transporters that are actually responsible of distributing the glucose to between the different tissues. We have GLUT1 transporters for the mammary gland, which is really low in the km value, making sure that the mammary gland is prioritized highly above actually the uterus, which is doing the farrowing, for instance. And as you can see up here, the uptake of glucose from the GI tract is really changing with the postprandial period. So half an hour after feeding, we have a maximum influx of glucose coming into the portal blood. And as soon as it comes into the circulation, it is distributed to somewhere else. It goes really rapidly. It goes less rapidly when we come 7 hours after the feeding. So this is just some pictures illustrating that we did the surgeries. We put in 2 catheters in the mammary gland. We put in also catheter in an artery, and actually also in the uterine vein to understand what is the uterus doing. And then we took some blood samples, both from the artery and the mammary vein, and we infused the blood flow marker to measure what is the uptake. So above 0 here we have, this is the uptake of nutrients to the mammary gland 10 days before parturition, 3 days before parturition, 3 days after, and 17 days after. And below you see what is the release and the secretion from the mammary gland, released back to the blood in terms of carbon dioxide and also NEFA in late gestation, and after parturition, secretion, of course, also into milk. And what was really interesting to us in the late gestation, that was the fat retention in the mammary gland. It is really a lot of energy that is being stored in the mammary gland, approximately 130 grams, which is quite a lot. It's each day that happens. And yes, we do know that from other studies that the mammary epithelial cell, that fat is accumulating just before parturition, ready to be released in the colostrum, but we do not have any idea of the amounts, and it's clearly not 130 grams per day that is being stored there. It's probably also in the mammary fat pad, but clearly there is quite a lot of fat being stored in the mammary gland in late gestation, which is something we have to understand better, I would say. We did a similar study as what I've just described, but now we shifted completely to the colostrum period, measured what is the uptake of nutrients in the colostrum period, and what is being secreted or released back to the blood in the colostrum period. And you can see here there's a really nice 1-to-1 ratio between the uptake and the release or the secretion, meaning that the colostrum that is being secreted is actually produced just as it is being removed. If the colostrum was produced when when the first piglet was born, we would not have seen anything really above above 0 here, but we see this is as high as what is below the 0 here. What is also interesting is that if we feed more fiber, we can see that more of the there's some carbon we cannot account for, and I believe that's because we have enhanced the buildup of fat in the mammary gland before parturition, because fibers are taking up as short-chain fatty acids, and the only thing that can happen with that is either oxidation or retention. I believe this yellow part here is actually some extra retention of mammary fat. Changing into immunoglobulins, we're also interested in understanding when does that becomes transferred into the mammary gland. We did the same AV difference, so taking blood samples from the arterial and venous blood and multiplying with blood flow, but we could not detect an AV difference that was different from 0, so we couldn't really measure that with our model. And the reason for that is most likely because immunoglobulins are really big molecules, so it's very few molecules that are taken up as compared with glucose, which is a very small molecule. We could see though in the arterial blood that the concentration of IgG declines, and that really occurs during the last 10 or 14 days of gestation. And then it stays low during the farrowing. If we consider the growth factors in the colostrum, we're also interested in understanding when are they being transferred into the colostrum. We could also not detect an AV difference that were different from 0. So just by evaluating the arterial concentrations of the growth factors, we could not say, well, they are being depleted. It seems like they are being increased, and most likely it's just the very last few days. Maybe it is during the colostrum period they are transferred. We don't know it based on these basically. This is some modeling which has been done by my postdoc that was in France for a couple of years, and they did some modeling trying to understand how much is it actually accumulated in the mammary gland in between the suckling periods. And this is where we also consider colostrum cannot be produced in when the first piglet is born, because in the mammary gland we can have, depending on the model, between 30 to 80 grams per gland when it's completely filled. And if you multiply that with 14 glands, we would probably reach a total of maybe 1 kilogram of colostrum. But maybe you remember that a sow produces 6.4 kilogram of colostrum. So it is really being produced mainly as it is as a response to colostrum removal. Considering the transfer of Ig from the blood to the mammary gland, it seems like it happens during the last 10 days, and then it becomes completely removed, and it actually becomes diluted from suckling to suckling as the colostrum period is taking place. And this is actually pretty much draining the sows for immunoglobulins. So we're also interested in the farrowing process, because the colostrum production, when we do that, happens and coincides with the farrowing process. And a few years ago, we realized, when the sows are doing the farrowing, they are really running out of energy. So if a sow is unlucky to start the farrowing too late relative to the last meal, they're getting exhausted. They are running out of energy. And this is something we have to consider as well, because farrowing and colostrum production, that happens at the same time. For the sow, it really has to complete the marathon. I see it as a marathon, doing the farrowing and doing the colostrum production. It has to be completed before plasma glucose becomes critically low. And it is critically low if the farrowing starts too late, or if we don't feed them properly. And we have actually started to reconsider how should we feed the sow when she approached the farrowing. And we've done a dose response trial recently, where we can see a very nice response here in terms of extra feed that is reducing the farrowing length until a certain stage, and then it starts to increase again, because then suddenly we see some physical blockage of the birth canal due to too much undigested fiber in the intestine. But the interesting thing here is we saw an optimum of the farrowing process of 4.1 kilo, but when we considered the colostrum production, we could maximize that at 3.1 kilo, sorry, 3.0 kilo, according to our cubic model here. So this illustrates, as I said before, colostrum is being prioritized above other things, mainly due to these GLUT1 transporters, probably because glucose and energy is a limited resource in these sows. Yeah, so talking about colostrum, we really have to have a good start of life. We need vital piglets. We need a high quantity of colostrum. We need a high quality of colostrum. Good start of piglet life starts with a high birth weight, which is reinforced with a high colostrum intake that will give a high survival rate. And the colostrum induces stimulation of intestinal growth and growth of other body organs. And that will lead to increased weaning weight gain in the pre-weaning periods, which increases the weaning weight, and it actually also stimulates growth post-weaning. But if you have a bad start of life, everything becomes negative. So weak piglets means low quantity, low quality of milk, and ultimately the survival rate drops and the pig may end up dying. So I really see the neonatal life is being kickstarted with the colostrum, and energy in the colostrum is important for the short-term survival. Growth factors are important for stimulating growth and development, and immunoglobulins are important not for the short-term survival, but more for the longer-term survival, at least until weaning, and maybe also the next couple of weeks until their immune system is fully developed. And just to illustrate my final slide for the impact of the colostrum on the piglet performance, you can see those piglets that are consuming relatively low amounts, they are being weaned at 6 kilo. Those that are consuming high amounts, that are weaned at nearly 10 kilogram. And this is when we adjust for the impact of the birth weight. We can see that for each gram of extra colostrum, we can increase the piglet weight at weaning by 7.7 grams. So this is really something that has a profound effect on the performance of the piglets. So just to conclude from my talk here, I would say suckling that is really important for inducing the mammary development and maintaining lactation function, including the lactose synthesis. In the mammary gland, we see quite a lot of production of lactose and fat after the first piglet is born, not much before. Some of the fat is being produced or stored in the mammary gland before, but it seems like lactose is nearly only produced after. IgG are being transferred before. We don't know really when the growth factors are being transformed and transferred to the colostrum, and that's clearly something we would like to understand better. Colostrum is being prioritized above the farrowing, and colostrum synthesis is really tightly coupled to the farrowing process, and this is something that has changed our vision in the last few years, I would say. Colostrum is highly important for piglet survival, and it really kickstarts the life of the piglets. So with that, I would like to say thanks very much for your attention, and I'll be happy to take any questions.